Welcome everyone. In this poster, we wanted to introduce use of growth phenotypic data from diverse biological systems to discover and validate new protein functions. I'm Zahmeeth Sakkaff, working as postdoc at Argonne National Lab, and I'm working with Pamela Wiesel, Chris Henry, and Jim Davis from the same lab. The reason we wanted to introduce this work is accurate functional annotation of protein in microbial genomes remains a challenge in biology. This grows with the assembly of hundreds and thousands of new assembled genomes every year. New genomes contain numerous unannotated proteins that have never been characterized in the lab. The current effort has limitations such as limited data, existing data is disorganized, poorly described, and difficult in mapping to the genome sequence of the species with experiments. Apart from that, obtaining new data are laborious and scale poorly compared with the amount of sequence data available today. Although numerous automated algorithms exist today for annotating the proteins, we are adopting new computational methods such as machine learning, combined with mechanistic insight from the metabolic modeling. This generates new data as well as predictive models with computation to have insight to the biological questions. The motivation is modeling. We develop new modeling to predict growth phenotypics for microbes and to improve the metabolic models. Also, machine learning will provide growth phenotypic predictions that may be used to improve the metabolic model's accuracy. As you can see in this below diagram, experimental growth data can be utilized in the mechanistics and machine learning models to build predictive models, and then we can compare their performance. So we started with collecting growth phenotypic data, especially biolog data from various papers and environmental isolates from the collaborators. So we were able to curate 170 genomes with growth predictions for 64 carbon sources. Machine learning model, we use different classifiers using scikit, especially logistic regression. So we develop predictive growth on each carbon source based on Patrick annotations as a features. Same way, we built the metabolic modeling. We applied model seed pipeline for each genomes, and we use the same annotation, and everything is built in K-base environment. As you can see, in this slide, we are comparing the accuracy of machine learning with metabolic modeling. So the first plot shows the per carbon source, and the second plot shows the per genomes. And the next x-axis explain about the substrates labels. And then here in the other plot, it shows all the genome IDs, and we are providing false positive, false negative, and correct predictions on the y-axis. So the performance here is put it on the table, which is easy to describe. Average correct prediction accuracy for metabolic model genomes, for machine learning models genomes and metabolic substrate is 74%. For metabolic model genomes 58, and for metabolic substrate it is 60. And also we count the number of correct prediction compare with the metabolic model and machine learning. We see the machine learning has 137 genomes and substrate 56 models. The same way, the metabolic model accuracy, which is showing higher in compared to the machine learning for correct prediction, 34 genome models and 7 substrates. Also, the equal models are like for genomes 7 and for substrate it's 1. So the takeaway is machine learning model shows higher overall correction predictions with erroneous predictions being evenly balanced between false positive and false negative. In contrast, metabolic model had a lower overall accuracy and display fewer false positive and far more false negatives. This is because the vulnerability of mechanistic model to missing genomes annotations. For example, one missing annotation result in a negative prediction in a metabolic model. As a conclusion, the machine learning model performance are high compared to the metabolic model, and machine learning model will improve the existing practices in the field. And also combining both will provide deeper understanding of biological system, especially top features, most influential substrate will predict what metabolic pathways are prominent in each substrate. Also, as a next step, fitting models to machine learning improve the accuracy of metabolic model, which we wanted to try, and also implementing to test combining machine learning and ML to a large set of experimental data to address protein annotation. Finally, we wanted to report most influential substrates for what to test with wet lab experiments. Thank you for your attention, and it's open for the questions.